Hello. The first part of this lecture did not have audio recording, so I'm going ahead to re-record the audio for this lecture, or for the beginning section of this lecture right now. Fortunately, this is also a very chalkboard heavy section of the lecture, so I'll be able to draw out some of this section on my iPad. I apologize if this doesn't match your notes perfectly. Before this point in the semester, we talked about barrier defenses a little bit, as well as intrinsic immunity, and we'll come back to the barrier defenses a little bit later in the semester. And we've spent the last few class periods talking about innate immunity. We're now going to move into thinking about some aspects of adaptive immunity. And in starting to talk about adaptive immunity, we're going to discuss a set of experiments that some early immunologists did that gave them some insights into adaptive immunity. So if you recall, at first, when a pathogen invades the tissue and may start to proliferate, we might see the immediate innate immune response during the first 4 hours or so. This is a situation where the pathogen will be recognized by things that are already formed. This might be our complement proteins or other antimicrobial proteins. The key thing here is that we have these preformed proteins. Sometimes this will be enough and the pathogen will be eliminated, the infection will end. Other times, however, the pathogen will not be eliminated by those preformed proteins, and next our innate immune cells will start to respond. This might be our macrophages and neutrophils as part of that inflammatory response that we've been discussing. And this is going to be something that has a little bit of a delay. It takes a little while for the cells to traffic and for the cells to actually become activated, do some transcription and translation. But within, say, the first 4 hours to 4 days following infection, these parts of the response will work to eliminate the pathogen. They may work to eliminate the pathogen, or they may not. And if they don't, then our adaptive response will start to be important. Usually we think of this adaptive response as only starting to come into play at about 96 hours following infection. What this should tell you is that we are again going to need some events to happen, some cells to move, some transcription, some translation, all of those types of things before we're going to have that adaptive immune response activated. The experiments that I want to tell you about are a set of experiments that were done by some immunologists named Van Bering and Kitasato in the 1890s, where they were looking at the immune responses, as you'll see, to some bacterial pathogens. I'm going to sort of combine the outcomes of a few different experiments as I describe this. So if you decide that you were going to try to look up the paper here, in fact, there are multiple papers that really are talking about the experiments that I am describing. So Van Bering and Kitasato did experiments, as I mentioned, with 3 different bacterial pathogens. One of those bacterial pathogens is Corynebacterium diphtheriae. That C is for Corynebacterium, which is the causative agent of diphtheria. They also did some experiments with Clostridium tetani, which is the causative agent of tetanus, as well as Clostridium botulinum, which is the causative agent of botulism. I'm only going to talk about the experiments with the 2 species of Clostridia, Clostridium tetani and Clostridium botulinum, because they are, of course, the most closely related. They are two members of the same genus, but no matter which bacteria we are looking at, we see the same kinds of results. And so we'll start with thinking about Clostridium tetani. First, these experimenters gave some mice a low dose of Clostridium tetani. This is a syringe injecting the mouse with Clostridium tetani. And specifically, this is a low dose. When they did that infection, the mouse lived. I'm using a mouse here because I know that I did that when I gave this actual lecture. As I mentioned during lecture, as I thought better of myself, I'm pretty sure they actually did this experiment with rabbits, but we're going to stick with mice. They then did a similar experiment where they infected mice with Clostridium tetani. But now with a high dose. And when they did that infection, the mouse died. From its Clostridium infection. They next did an additional experiment where they took a mouse that received a Clostridium tetani infection, specifically one with a low dose. Remember, this is an infection that the mouse lived through previously. And then they infected that same mouse again with Clostridium tetani, the high dose. And this is the dose that previously killed the mouse. However, if the mouse had previously received a low dose of Clostridium tetani before receiving that high dose of Clostridium tetani, now this mouse lived. And so this indicated to von Behring and Kitasato that previous experience with this infection somehow gave the mouse immunity to what was previously a lethal infection. So this lower dose of Clostridium tetani was able to provide some immunity. They also were able to see that this immunity was specific by doing experiments like one where they used Clostridium botulinum at a low dose, and then Clostridium tetani at a high dose. And in this case, the mouse would die. The first infection that the mouse received needed to be with the same microbe as the high dose that was sort of challenging the mice that could potentially be leading to the mouse's death. So this showed them this important immunological principle of specificity. You can see a slide talking a little bit about specificity here, that adaptive immune molecules like the antibodies that are Y-shaped proteins shown here will bind to one microbe specifically and not to another microbe. Here you can see measles versus influenza, showing this adaptive immune principle of specificity. And that was the same thing that von Behring and Kitasato saw. It wasn't as though the mouse's entire response to all microbes had improved after a low-dose infection. Instead, they only had a specific improvement of the microbe that they had already been challenged with. So now, von Behring and Kitasato went ahead to do their next experiment. In the next experiment, they again used a mouse. We'll again say that this was a mouse that was infected with Clostridium tetani at a low dose. And they wanted to understand what this immune response was that was protective. At this point in time, there was a debate in immunology about the difference between cellular and humoral immunity, was the important protective factor a cell, or was it something in the liquid of the blood, which they were thinking of as the humors? We now know that that would be some kind of protein. And so they took this mouse that had been previously infected with Clostridium tetani at a low dose, and they isolated 2 different samples from this mouse. One sample was the spleen, just like you did in lab 2, where you isolated spleens from a mouse. Von Behring and Kitasato isolated the spleen from these mice, trying to see if these protective factors from this mouse were in the spleen. Alternatively, they took the serum. The serum, as you can see here, is actually the liquid part of the blood that you can get if you let the blood clot, and you'll remove all of the cells, and you'll just end up with the liquid part of the blood, as you can see here, which contains all of the secreted proteins, including, as we now know, antibodies. So they separated out the serum to look to see if the liquid part of the blood had this protective factor versus the spleen to see if the cells had this protective factor. One thing I will let you know is that this experiment worked out very cleanly for them. They won the Nobel Prize for this experiment. However, we now know that they lucked out a bit in the 3 bacteria that they chose. If they had chosen some other types of pathogens, they would have gotten a different answer here. So I don't want you to take this as it's always the cells or it's always the serum. That is important. We now know that they were sort of lucky with the bacteria they picked, but this did help them start to understand what was going on. So they took either this serum from the Clostridium tetani exposed mice or the spleen from these exposed mice and transferred them into new mice. This is known as an adoptive transfer experiment. Immunologists do stuff like this all the time. Now these mice had either the cells or the serum from the protected mice. And these mice were then challenged with the high dose of Clostridium tetani. The mice who had received the spleen cells died, while the mice who received the serum were protected. And this told them that there was some kind of protective factor in the liquid of the blood. We now know, of course, that this is antibodies, which are a protein that are secreted in the liquid of the blood, but some further experiments that I'll be telling you about will allow us to see how they learned that these were the antibodies. As I said, we saw the principle of specificity with these experiments. Now, you might look at these experiments and say, oh, yay, she showed us an experiment from 1890. How fascinating. But in reality, we still use this kind of process in a number of situations. So there are a lot of situations where we will take antibodies from someone who has already been infected with something and transfer them into another individual to allow that person to have antibodies. The idea here is that we need to get antibodies into this recipient really quickly. We don't have enough time to let their adaptive immune response do its thing. And so we take antibodies from another individual, we put them into this person to protect them right away, just like was done with the mice in that experiment. This is known as passive immunity or passive antibody therapy. If, for example, you were to be bitten by a black widow spider, we would give you passive antibodies against that venom, because that venom is going to affect you in hours. You don't have time for the days of making your own antibodies. And you can see a few other situations where we use passive antibody therapy, even now. This was also the first therapy that we had for COVID. So at the very beginning of the epidemic, we actually had COVID patients, and we would draw blood to get antibodies. Again, just like what was going on in the experiments I just showed you, and give that antibody to others who were really sick to help them recover. This is very much something that is still going on. You may also have heard about some people receiving monoclonals or passive antibody or antibody therapies. Because what people basically figured out how to do is to make those antibodies in the lab instead of taking them from a patient, and put antibodies against the virus into patients as well. So you might have heard when Trump got antibody treatment, he got bamlanivimab. Physicians all call it Bam Bam because they can't pronounce it either. Which is just an antibody made in the lab to do this exact same process. He got 8 grams of it, which I don't know what that means to you, but actually after I read that in the newspaper or online, I went to the lab and measured out 8 grams of something, so I could have a visual representation of 8 grams, and it was shocking. So anyway, passive antibody therapy, while is something that we're still using now, even though it feels somewhat old-timey. People then did more experiments based on the experiments that von Behring and Kitasato did. Basically, what they tried to do was they tried to take this serum and divide it up more to try to figure out what is the thing in this mixture that is the protective thing. The first thing that they did was they treated it with protease, or lipase, or glycosidase, or nuclease, to see, is it a protein, is it a lipid, is it a nucleic acid, is it a carbohydrate? And they figured out it was a protein pretty quick. And then they decided they wanted to try to figure out what protein it was. They did this kind of experiment, which is, you can actually see this is from Tiselius and Cabot in 1939. This is like a precursor to a gel. So basically they could find that they could separate all of the proteins in the blood into 4 groups. In no way are these super pure. And you can see that they have albumin, and then they had these other ones that were just sort of proteins that were floating around. They called them the globulins. And they said one's alpha and one's beta and one's gamma. When they did further experimentation, they found that all of the protective ability was in the gamma globulin fraction. In fact, if they got rid of gamma globulin, then this whole thing no longer worked. So they were basically trying to narrow down which protein in the blood was protective. There are 2 reasons that I tell you this. One is that if you go into the clinic for a number of different conditions, we still treat patients with gamma globulin infusion, which just means we give them antibodies. But you will see clinically the phrase gamma globulin infusion. This is sort of where they got to that from. They also referred to gamma globulin as the immune globulin. And in general, we still call antibodies immunoglobulins based on that. So immunoglobulin is a very frequently used term for antibodies, or as I have heard people jokingly call themselves immunogoblins, but that's a different thing. That's more around Halloween. So further, biologists tried to study that gamma globulin fraction. And they found that it contained these proteins that we now know of as antibodies. And you can see a basic picture of an antibody here, that it was a Y-shaped protein. Here's an antibody. And in fact, you can even see antibodies under electron microscopes here, and that's really exactly what they look like, is this nice Y-shaped protein. A big part of what we're going to be talking about today has to do with what the parts of the antibody are, sort of zooming in on the structure and learning about different aspects of that structure. You can, in fact, see that in this figure from your textbook here, that we have lots of different parts of the antibody that are labeled and named in different ways. In general, we think about the sort of two top arms of the Y and the tail as being two separate things. In order to fully be able to tell you about some aspects of the structure of antibodies, particularly the arms, the Y parts of the arms, I have to take a little diversion and tell you some things about what antibodies bind. We're going to sort of talk about that, and then we're going to come back to the antibody itself. So the thing that the antibodies bind, officially, this is actually a molecule recognized by the adaptive immune response. So when we think about T-cell recognition, we'll use this same term. The molecule that antibodies bind is called an antigen. It generates antibodies. That's how it got the name antigen. And so the molecule that's recognized by the adaptive immune response is our antigen. Here you can see my antigen that can attach to my antibody. In the case of antibodies, the antigen can be anything. The antigen can be a protein. The antigen can be a lipid. The antigen can be a nucleic acid. The antigen can be a carbohydrate. The antigen could be almost anything. Really pretty much anything. In fact, in 1900, Landsteinner did some experiments where he was doing fancy organic chemistry and made chemicals that had never before been made. They were different dyes. And he injected those dyes into mice. So these are not proteins, not carbohydrates, not lipids, not nucleic acids. They're like synthetic molecules that had never before, the mouse had never before seen them, obviously, because Landsteinner was the first person to make them. And then he looked to see whether the mice made antibodies against these compounds. And he found that they did. So in fact, this showed that you could actually get antibodies against basically anything, like literally anything. Not only did he see that, but if you notice, the 4 different compounds he made had a relatively small difference between their chemical structures. So you can see this carboxylic acid here, and it's in the O, M, or P position, or not there. So it's not like a giant change. Mice that got this one only made antibodies against that one, and not against these other very similar chemicals. Same thing. Each mouse only made antibodies against that specific chemical that it was given. So we see 2 things here. One, another demonstration of the massive specificity of the adaptive immune system. And 2, the fact that we can get antibody responses against anything. But when we think about antibodies and antigens, There are some things you need to know about antigens. As I'm going to sort of draw some things out and explain some stuff, and hopefully as part of this description, you will also be able to say very smart things to your friends about various things that are in the news about COVID, because there are so many places where people mess up the next couple of things in the news, and so you'll know it and you'll be able to, actually, this is how it works. So antigens might be some kind of whole big protein. Here it's this whole little square. But we can divide up antigens. And we can think about the specific points on the molecule. The example that I'm showing here is a protein, and I will be pretty much using protein examples throughout today, but do remember this could be something other than a protein as well. Those proteins have sections, have different little pieces to them. And the antibody doesn't bind to the whole protein. It binds to a particular part of it. And that specific part that actually makes contact with the antibody is known as the epitope. What you can notice in this image is that our one antigen has two totally different epitopes. It has this blue epitope with these three little humps, and it has this red epitope with these two little humps. And so two totally different antibodies can bind to this antigen because they bind to different epitopes. One thing that is important about the epitopes that are recognized by antibodies is that those epitopes are generally known as native epitopes. You'll see some more about this on the next slide as well. But the idea here is that, again, we're looking at a protein antigen. The epitope, in the case of an antibody, is some part of the protein that's present in the 3D structure of that protein, perhaps in the tertiary structure or quaternary structure, when that protein is all folded up. Sometimes that might be something like this that you see on the left, where you have some amino acids that are consecutive, and they're consecutive in the primary structure when the protein is unfolded, as well as in the tertiary structure when the protein is folded. Other times, the epitope is made of amino acids that are only next to each other when the protein is folded. And when the protein unfolds, those are never going to be next to each other again, and we won't have this work. And so we often will see antigens that are only present when the protein is folded. And you can see this here as well. So in many cases, we might have an epitope that is only formed when our protein is folded into its tertiary structure. If you unfold that protein, you may lose that epitope and your antibody may no longer bind. There are other situations where the epitope may be some continuous amino acids, and so if you unfold the protein, then that epitope will not be destroyed. There are even situations where there are sort of epitopes that are hidden in the middle of the protein that are only present if you unfold the protein. You may look at all this and you may say, I am over immunology. I'm never going to do immunology ever again. But you might, if you are involved in the biological sciences, do a Western blot. Or you might do immunofluorescence. Or you might use a COVID rapid test. All of those things involve antibodies binding to antigens. And if you don't prepare your sample correctly, maybe you denature your sample when you weren't supposed to, you might mess up you might ruin the whole thing. If you're in this situation and you need to not denature your antigen, and you do, you just ruined it. Antibody can't bind anymore. You're gonna get false negative. So this piece of information is actually really important throughout most of biomedical sciences, even if we're not in a situation that is sort of specific to immunology. So the part of this that is a little bit challenging for people is that, as I mentioned before, antigens can contain multiple epitopes. Here you see two different examples of antigens that contain multiple epitopes. Here you can see an antigen that has 4 different epitopes. Here you can see an antigen that has the same epitope repeated over and over. In either case, multiple antibody molecules can bind to those antigens. Here it's 4 different antibodies binding. Over there it's 2 of the same antibody binding. But in the end you can see multiple antibodies can bind. In addition, when we actually look at a microbe could contain multiple antigens. So here you can see a bacterium, it's got tons of different proteins on it, multiple antigens. And if you put all of this together and actually think about a real microbe, you get something that looks a little bit like this. So on the left, you see a cartoon version of HIV. I show HIV, 1, because it's one that I spent a whole lot of time thinking about, and 2, because it's a relatively small microbe, has a relatively small number of proteins. You can see its proteins here all labeled. All of those proteins could be different antigens. If we zoom in on just one of them, which is this GP120 thing that you can see here in yellow, you come over here. These are all different epitopes that people have mapped in just GP120. And we could go through every other part of this virus and map epitopes. So I mention this because it's what you should hopefully realize is it's not like, sometimes I'll say, and this is what happens with your antibody against HIV. You don't have one antibody against HIV. You have one antibody against this epitope and 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 all the epitopes and all the epitopes of all the other proteins too. That of course is sort of a thing to understand because we see the same kind of thing with SARS-CoV-2, which has multiple different proteins in it, each of which is an antigen, and we can take one of those antigens, like the spike protein that's found in the vaccine, and we can actually zoom in and start to identify lots of different epitopes. Here they're showing you, this is at the beginning when they were kind of doing the first 6. More recently, people have actually shown that just in the spike protein of SARS-CoV-2, there are at least 20 antibody epitopes. So you don't have one antibody that binds to SARS-CoV-2. Spike. You have 20. And so when people are like, oh, this variant escapes, this variant does this, well, it might escape one of the 20. And oftentimes some of the reports are about, oh, it escapes one of the 20. And the press is like, it means it escapes and we're all going to die. But in reality, it escapes one of 20, but the other 19 are still fine. One antibody is not going to work anymore, but the other 19 work. So understanding the fact that there are multiple epitopes is really important. Understanding that there are multiple antigens in a microbe is also really important. If you go to, and I say this, I have actually had physicians write me emails about this next point because they were confused. Because I do podcasting, and so they knew of me from the podcast, and they're like, I have this terrible problem, you need to solve it. I'm like, this is pretty basic immunology, guys. SARS-CoV-2 has multiple antigens as well as multiple epitopes. If you go down the road to Quest and have your antibodies tested to see if do you have antibodies against SARS-CoV-2, they only check for antibodies against one antigen. They don't check for antibodies against every single antigen. Specifically, they're checking for antibodies against N to see if you made an antibody response. And if you're infected, you definitely have antibodies against N. Here's the thing. The vaccine only includes S. So if you got vaccinated and didn't get infected, you have no antibodies against N. You only have antibodies against S because they're different antigens. Yeah, like I said, I have had people writing me emails. I got vaccinated and then I went and got an antibody test and it was negative and I don't know what's going on. I'm like, yeah, it's because you've got a test against the wrong antigen. So realize that there are multiple antigens, multiple epitopes here, and this actually explains a shocking number of questions that people have on this topic. One other thing that I've sort of implied as I've been talking, and it also shows up in the title of this paper, is that when we look at true, real antibody responses in people, we see that those antibody responses are polyclonal. And so you can see a polyclonal response on the left here. What that means is that if I immunize you with a particular antigen, here you can see one being immunized with myoglobin, you're going to make many different antibodies. You're going to make antibodies against different epitopes. Here you can see you can make the red antibody against the triangle epitope, the blue antibody against the square epitope, and the black antibody against the whatever half circle epitope that is, epitope. So you're going to make multiple, just like and if we actually then had multiple antigens, it's even more complicated. So, I can't say your antibody against HIV, your antibody against SARS-CoV-2, because there's really this whole mixture that your immune system has made of antibodies against different antigens and in different epitopes on those antigens. Sometimes in the lab, we have the ability to make antibodies that are pure and that are only antibodies against one epitope, and usually one epitope on one antigen. That is not what we see in a normal organism. That's something that we have to do a fair amount of lab manipulation to make. Those are called monoclonal antibodies. We use them in a lot of techniques. We use them in the clinic in a lot of different places. So oftentimes I think people imagine antibodies as monoclonal, and often the ones that we buy and use for our experiments are monoclonal, but the antibody response that you would make against a particular antigen would be a polyclonal response. So, the thing that is binding to our antigen is our antibody. One thing that is important about antibodies is that our antibodies have these two arms that we're going to be talking more about. And these two arms, this region, is actually is the region that is important for, as you will see coming forward, binding to antigen. And there are two of them, and they're identical. And so we also have to think briefly about why it matters that there are two of them and that they are identical. If we just had one arm, we could see sort of an on and an off rate of basic biochemistry.
And that would tell us things about binding strength. Woohoo. Because we have 2 arms, we get a different binding strength. And you would normally be like, yeah, great job, Captain Obvious. It's twice as much as the other one. But it's not really twice as much as the other one. It's actually more than twice as much. And the reason why it's more than twice as much is because these 2 arms are close together. And if one of them comes off, this other one is held really close. And so it's pushed to come back to bind. It's not actually fully able to go away and leave. So it's not actually totally in solution. It actually is favorable for it to come back and bind again. And so you end up getting an even higher binding strength because of these 2 identical arms, to be able to bind to 2 identical copies of the epitope. So instead of sort of going on and off, the antibodies kind of sort of bounce but stay on that antigen. We refer to that as getting some increased avidity of the antibody. One other piece of information I have to tell you about antibodies. I'm kind of right now hating the order of a couple of these slides, but that's okay. I'm going to roll with it. Is that there's one other thing you just generally I want you to know about antibodies, and part of the reason I'm telling you now is because I am sure that I will slip and say this, because I've been thinking about this stuff for a long time, so I want you to just be aware of it. Antibodies actually do come from cells. We often talk about that whole cells versus proteins distinction in the immune system. But antibodies do actually come from cells. Specifically, those antibodies come from a type of lymphocyte called a B cell. And sometimes even from a specific type of B cell called a plasma cell. So plasma cells are like the B cells that are extra amazing at making antibodies. Other B cells can make antibodies. They make a little. Plasma cells make so much. So as we talk about the antibody and its structure, and next week when we talk about how we generate that, sometimes we're talking about what the B cell does to make the antibody. And so I may just sort of slip and be like, well, and then the B cell. And so I want you to realize how the B cell is connected here. The other place where that becomes a little bit slippery is that if you look at a B cell that is making an antibody, so here you can see these B cells making antibodies. Those B cells also have a receptor on their cell surface. Again, immunologists, so creative. The name of the receptor on the B cell surface is the B cell receptor. And if you look at this image from your textbook, you'll notice that the B cell receptors look kind of similar to antibodies. And in reality, the B cell receptor is an antibody with a transmembrane domain. It actually is an antibody. They're made, the B cell receptor and the antibody are made basically together and in the same kind of way. And so if I slip and suddenly start talking about the B cell receptor, I actually mean the antibody, because they're the same thing. So if I mess up, that's the background of what's going on, because in my mind, in a lot of ways, these things are interchangeable. Okay, now to this, which I feel like is what I wanted to be talking about, and those last things were in a weird order. We can sort of zoom in more closely on this Y-shaped protein and figure out what we know about its structure. And again, scientists in the past did this to try to figure out what was up. And the first thing that they did was they took the antibody and they treated it with a chemical called mercaptoethanol. Mercaptoethanol is a reducing agent. It breaks disulfide bonds. And so when they treated the antibody with mercaptoethanol, they found that they actually got 4 proteins. 4 protein chains. And that told them that the antibody is made up of 4 separate protein chains that are held together by disulfide bonds. When they looked more closely, they saw that 2 of them were kind of big, 2 of the 4 proteins, and 2 of them were kind of small. They called the big ones heavy chains. They called the little ones light chains. And the 2 heavy chains from any particular antibody were identical. And the 2 light chains from any particular antibody were identical. So this Y-shaped protein is made up of 4 chains, 2 identical heavy chains, which are shown in this one in yellow, and 2 identical light chains that are shown in orange. They also used some different proteases to try to understand the structure of the antibody. One of them was a protease known as papain. You guys actually know some things about papain, even though you've probably never heard of it in a biochemistry kind of way, and it's not like a famous protease in biochemistry, but it is a famous protease actually in your life. So imagine for a second that you are eating some papaya, papain, because papain comes from papaya, not the other way around, or pineapples. If you eat either of those fruits, is there anything weird that happens to your mouth? See some yes head shakes. What happens, Delaney?
Your tongue is itchy.
You get a little itchy. You sort of have this weird like tingly feeling sometimes. Yeah, that's because those fruits contain this protease that's starting to act a little bit in your mouth. That protease is called papain. And so they used this protease that people cared about back in the day. Now people only care about it because of papayas. And they cut up the antibody. And when they did their papain digestion, they got the antibody in 3 different parts. You can see those 3 parts here. There's one thing I forgot to tell you on the previous slide. When they did their mercaptoethanol and they got the 4 separate chains, if they tried to take this antibody and bind it to antigen again, it didn't work. So you can't, none of these products bind antigen. I tell you that because that's not the same thing that you see in this case. Here we see that there are these 2 identical pieces that each contain a light chain and part of a heavy chain. And this other piece that contains the rest of the heavy chain. And what they found is that these little pieces still had the ability to bind antigen. This part that had the light chain and this piece of the heavy chain still could bind antigen. They in fact named this piece the Fab fragment. You can see it from your textbook, the Fab fragment as well. And the name of the Fab fragment was the fragment that had antigen binding. So this could bind antigen, so it was the fragment with antigen binding, and they called it the Fab fragment. They also found this other piece, sort of the tail. They called it the FC portion. This is not what FC originally stood for, but I'm going to just say it because it makes your life easier. You can think about that as being the constant portion, the fraction that is constant. And so they found that the antibody had these 2 identical arms that could bind to antigen. Same antigen on either side, totally identical, FAB arms, and then we've got the FC tail. They did also do a digestion with pepsin, and when they did a pepsin digestion, they basically got one fragment, which was this fragment that you see here, the Fab2, which has the 2 Fab arms still held together by their disulfide bonds, and the rest of the Fc was completely degraded. And so one of the things that they were able to find out was this aspect of the structure of the antibody. So they could see that any antibody has two identical heavy chains held together by these two disulfide bonds right here, as well as two identical light chains. And there's a light chain held to a heavy chain with one disulfide bond as shown there. And so you can see the light chains in yellow, the heavy chains in green, the disulfide bonds in black. This is the general antibody structure. Yay. You will also notice that on this image, as well as many of the other images that people always show for antibodies, we often draw the antibody as being made up of these little boxes. And here these boxes are touching. They're actually covalently linked together. Here you can see there is the linkage a little easier. But we've got these little boxes that are often shown. Those boxes are depicting a particular type of protein fold. This protein fold was first discovered in antibody proteins, and it is called the immunoglobulin fold, or an immunoglobulin domain. Remember how I told you we sometimes call antibodies immune globulins or immunoglobulins? Yeah, so we named this fold after them because they were where it was discovered. And this is an example of an immunoglobulin fold here. You can also see an example of an immunoglobulin fold in this image from your textbook. There are a couple of things I want to point out about the immunoglobulin fold. So the immunoglobulin fold contains multiple beta sheets, which are shown by these arrows, held together by some loops. And so if you look at this one that's on the left, you can see we've got, we start, there's the N terminus, here I'm starting, the protein goes this way, and then there's a loop, and it goes to the next beta sheet, and then there's a loop, and it goes another one, and then there's a loop, and it goes another one, and then beta sheet and loop, and so you can kind of see it's all this big connection of these beta sheets with these loops. There's also a disulfide bond in the middle that sort of folds the whole thing over. When I was originally taught about this, I was told that it looked like one of those little sandwiches with a toothpick in the middle. The disulfide bond was the toothpick, and here's the sort of the sandwich from the side. We are going to care a bunch about some of the little loops that you can see in the immunoglobulin domain. So notice that when we make that immunoglobulin domain kind of fold up like a sandwich, there are 3 loops on top. Sort of come off like 3 little fingers at the top. We're going to care about those a little bit later. So when I come back to with the loops in the immunoglobulin domain, that's the loops in it. You can also see this in this image from your textbook that's really kind of showing us the same thing. And I will also just tell you that in immunology, so many important proteins have immunoglobulin folds. It's like the major protein fold we see in immunology. Some textbooks draw it like this little circular thing with a disulfide bond. Sometimes when I draw it on the board, I will also draw it that way. And so this is just showing you a sample of proteins in the immune system. And what you should notice is they're all immunoglobulin folds. So this is sort of a key part of the structure. So I've got to tell you a few other things about aspects of antibody structures. I mentioned the difference between the FAB, the part that binds to antigen, and the FC, the part that is constant. We are going to spend a whole lot of time at some point talking about how we get differences in the parts that bind antigen, which are known as the variable regions. And so you can see that the very last immunoglobulin domain of the heavy chain and the light chain that is binding to antigen is known as the variable region. Shockingly, it varies a lot. There are millions and millions and millions and millions and millions of different kinds of them. You can also see that we have this other part of the antibody. It includes both part of the heavy and light chain, includes sort of the end of the Fab, as well as the FC, and that region is constant. That's known as the constant region. And the constant thing here is sort of relative. There are a few different kinds of constant regions. For right now, I'm going to say that there are 5. Some of these do have subtypes. So IgG has subtypes. I'm not going to focus a ton on the subtypes right now. I'm just going to say that there's 5 different types. So you can see that there are 5 different types of constant regions. So it's not like 100% constant, but it's not millions. It's only 5. They're called IgM, IgD, IgG, IgE, and IgA. Or you might have heard the phrase, an apple a day keeps the doctor away. I remember them as MDs give everyone apples. To remember the 5 different types. And so these different antibodies that have the different types of constant regions are known as different isotypes. We'll be talking about flow cytometry tomorrow, which is a technique that uses antibodies. So be on the lookout for an email from me, but you will likely need computers tomorrow. And then we will continue talking about aspects of antibody structure, as well as how in the world a little bitty protein can do so much stuff against a microbe on Friday. So I will see you guys in lab tomorrow.